Ophiocordyceps unilateralis, the 99-million-year-old mind control fungus. Picture an ant, not dead, not sick, not stumbling, walking with absolute purpose. It climbs a plant stem, stops at a very specific height, locks its jaws into the leaf tissue with a bite so precise it leaves a geometrically perfect scar, and then it waits to die. It has no choice in any of this. Something else is steering. Something that has been doing exactly this to the same depth at the same height with the same mechanical precision for at least 99 million years. Before the asteroid, before the extinction, before the world that produced us had any idea what was coming. In 2020, a team led by researchers at the Nanjing Institute of Geology and Paleontology announced a discovery that quietly rewrote the timeline of biological horror. A piece of amber from Myanmar, fossilized tree resin from the mid-Cretaceous period, contained an ant with a fungal stalk erupting from its body. The amber was 99 million years old. The fungus preserved inside it was Ophiocordyceps, the same genus doing the same thing in tropical forests today. 34 million years before the dinosaurs ended, this system was already operational, already refined, already perfect. What makes it perfect is the specificity. This is not a fungus that simply kills its host. Any parasite can kill. Ophiocordyceps controls. David Hughes of Penn State University spent years measuring where infected ants die in Thai rainforests, and what he found borders on the uncanny. Infected ants consistently bite into vegetation at approximately 25 centimeters above the forest floor. The variance is less than 2 centimeters across hundreds of measured specimens. That height is not random. It corresponds precisely to the microclimate zone of optimal humidity for Ophiocordyceps spore release, warm enough to sporulate, moist enough to disperse. The fungus has calibrated its behavior override to the specific weather of the forest floor. And it does this without a single neuron to measure anything. A 2010 study by Hughes and colleagues in Biology Letters pushed the record back further to 48 million years ago, finding fossilized leaves in the Messel deposit in Germany bearing the identical death grip bite scars that infected ants leave today. Submillimeter geometry. Identical across tens of millions of years and thousands of kilometers. Then, a 2017 paper published in PNAS by Fredrickson and colleagues revealed something even stranger about the mechanism itself. The fungus does not invade the ant's brain. It surrounds individual muscle fibers with high-fall threads and issues commands through the muscles directly, leaving the brain largely intact, physically present, chemically bypassed. The ant is not unconscious, it is overruled. There are over 200 known species of Ophiocordyceps. Each one evolved to control a specific host species, not a category of insects, a species. The pharmacological formula is tuned, and infected ants are expelled from their colony by healthy nestmates before the takeover completes, meaning the colony has developed a counter-response to a threat it has been fighting for tens of millions of years, a cold war between social insects and a fungus that has been running longer than any war in the fossil record. Evolutionary biologists describe this as extended phenotype. The parasite's genes expressing themselves through the host's body, shaped over geological time by the relentless pressure of selection. Those fungal lineages that pushed ants to slightly better spore release positions spread more offspring. Over millions of generations, the precision we see today accumulated the way any complex system accumulates. Incrementally, inevitably. But incremental accumulation does not explain how a eukaryote with no nervous system, no sensory organs, and no exposure to the forest floor during its own development acquired a pharmacological map of the ant's motor system precise enough to override it on first contact. The WNT signaling proteins, the circadian timing, the spatial calibration to a 2-centimeter humidity band, These are not rough approximations. They are solutions. And when you look at a 99-million-year-old amber specimen and see the same solution, unchanged, staring back at you, a reasonable person is allowed to ask, how long does it take to accidentally become that precise? And what exactly does it mean to know something without a brain to know it with? From a parasite that commandeers the nervous system while leaving the body intact, we descend to something that takes a different approach entirely. It does not borrow the body, it becomes a body part. Cymothoa exigua, the parasite that became a tongue. You are a snapper fish. You have a tongue, a functional, muscular organ that helps you capture prey, generate suction, and process food. You have had it your entire life. You will not notice when it begins to disappear. A small crustacean entered through your gill slit when it was still a juvenile, navigated the dark interior of your body in complete silence, found the base of your tongue, and began drinking the blood supply. Over the following weeks, the tongue atrophies, the blood is gone, the tissue withers, and the crustacean, now fully female, hooks her modified legs into the same muscles your tongue once used and takes up permanent residence. She fits so well that for years, marine biologists who found her in dissected fish assumed she was part of the fish's anatomy. This is Cymothoa exigua, the only known animal parasite on Earth that functionally replaces a host organ, not destroys it, replaces it. And she has reportedly been doing this since at least the early Cretaceous period, approximately 119 million years ago, when related parasitic isopods were already embedded in the bodies of fish swimming in ancient seas. The life cycle compounds the strangeness. Cymothoa begins as a male, swimming free until it locates a suitable host fish and enters through the gills. Once established inside, if no female is already present, it transitions to female. One of the relatively rare examples of sequential hermaphroditism triggered by a parasitic life cycle. The female then severs the lingual artery at the tongue's base, cutting off blood flow until the muscle dies. She attaches to the remaining stub using modified leg tip claws called dactyli, hooking into the same musculature the tongue formerly occupied. The fish continues eating, continues swallowing. The replacement is functionally adequate. Some studies have found no measurable difference in feeding efficiency between infected and uninfected fish. The morphological documentation of this attachment was detailed by Brusca and Gilligan in a 1983 study in Fishery Bulletin, describing how the isopod's leg structure physically mirrors the architecture of the tissue it replaced. Male subsequently enters the same host, fertilizes the now established female through the gill cavity, and the female broods eggs while still living inside the fish's mouth, raising the next generation inside the body of the animal whose tongue she became. The female can reach nearly 3 centimeters in length. The fish does not eject her. Cymothoa exigua has been confirmed on at least 8 fish species in the Gulf of California alone. A related cymothoidy specimen drew international headlines in 2005 when a consumer in the United Kingdom purchased a snapper at a supermarket and found the isopod still attached inside the fish's open mouth, apparently mistaking it initially for an oddly shaped part of the jaw. The image circulated widely. People were unsettled in a way that took a moment to articulate. Then it clicked. The thing inside the mouth was not a deformity. It was a tenon. Evolutionary biologists trace a plausible path from external gill-clinging parasite to internal organ replacer, ancestors that migrated deeper into the body over millions of generations, those that found blood-rich tissue surviving better, those that happened to anchor to muscle attachment points persisting longer. Selection does the rest. Over 100 million years, This ratchet produced what we see today. But the ratchet has to turn in very specific directions simultaneously. The parasite must sever the correct artery to destroy the tongue without killing the fish from hemorrhage. It must locate the muscular attachment stubs in an internal environment it cannot see. It must produce a body shape and attachment architecture that replicates tongue function well enough that the fish does not starve. Every intermediate stage between gill dweller and functional prosthetic tongue had to be viable enough to reproduce. Some parasitologists note that the number of simultaneously correct adaptations required is not a reason to reject evolution, but it is a reason to sit with the improbability for a moment longer than we usually do. Simathoa tells us that the boundary between self and parasite can dissolve so completely that the host no longer experiences the difference. The fish does not know its tongue is gone, it cannot know the replacement is too good, and that might be the most disturbing detail in the entire life cycle, not the theft, the seamlessness of it. From the ocean and its stolen anatomy, we move to the shoreline, where a parasite has found an even more total solution to the problem of control. It does not replace an organ, it replaces an identity. Saculina, the barnacle that erased a crab's identity. There is a crab on the seafloor that has forgotten it is a crab. It no longer grows new claws when old ones are lost. It no longer defends territory, competes for mates, or performs any of the behaviors that have defined its species for millions of years. Instead, it spends its days grooming a soft, pale lump on its abdomen, fanning water across it, turning it carefully, protecting it from disturbance with the attentiveness of a mother tending eggs. Those are not its eggs, they are not its anything. The crab's reproductive system was shut down months ago. Its hormones have been rewritten. Its behavioral identity has been fully overwritten by something that entered it as a microscopic mass of cells and has been slowly, methodically taking the whole animal apart from the inside. This is Saculina, a barnacle, and it represents what some biologists quietly describe as the most complete example of parasitic colonization in the animal kingdom. The life cycle begins with a free-swimming female larva called a kentrogon, which locates a crab, finds a soft joint in its exoskeleton, and injects a tiny mass of undifferentiated cells, essentially just raw biological material, directly into the crab's bloodstream. That mass then grows an internal root system called the interna, which threads through the crab's digestive gland, gonads, nervous tissue, and eye stalks over the course of several weeks. The crab's immune system mounts a response. The interna survives it. When the invasion is complete, a small external sac pushes through the crab's ventral surface in the exact location where a gravid female crab would carry her egg clutch. The crab begins tending it. Janhui's histological work, published in the Journal of the Marine Biological Association of the United Kingdom in 1995, traced the interna through dissected specimens and found it present in virtually every major organ system simultaneously. Not a partial infection, total occupancy. The crab continues to function, walking, feeding, responding to stimuli. But every metabolic resource is being redirected. The interna suppresses E-edisone, the molting hormone, permanently. The crab never molts again. If a claw was lost before infection, it would have regrown at the next molt. Post-infection, it does not. All cellular repair capacity now belongs to Saculina. The male follows separately. A second larva locates the external sac, enters a tiny pore in its surface, and migrates internally to fertilize the female. A complete reproductive event conducted entirely within the body of a third organism which has no awareness of it. Over 40% of shore crabs in some European coastal populations carry active Saculina infections. Related rhizocephalan barnacles have been identified in fossil crustacean material from Cretaceous-age deposits, placing this parasitic architecture well over 100 million years into the past. Evolutionary biologists explain the life cycle as the logical extreme of parasitic simplification. A barnacle that abandoned its shell, its legs, its filter-feeding apparatus. All energetically expensive structures irrelevant when a host provides everything, and retained only the capacity to infiltrate, grow, and reproduce. The behavioral feminization of male crabs is framed as a byproduct of hormonal disruption. The same signaling pathways that control molting also regulate sex-specific behavior, so suppressing them produces female brooding responses as a side effect of resource redirection. But a side effect does not explain the male larva's injection event. The female, already inside the crab, must release a chemical signal that the male detects from outside, through the crab's body wall, and uses to locate a specific microscopic pore in the external sac. This is not an overflow of a disrupted system. It is a coordinated two-organism reproductive act conducted through the body of an unwilling third party. The precision of that signal, the existence of that pore, the male larva's capacity to find it, none of these are incidental. They are the system. Some researchers studying Sacculina have noted that the parasite's long-term survival depends entirely on a healthy, mobile, feeding crab. In some measurable sense, Sacculina has more investment in the crab's continued well-being than the crab has in its own future. The crab is not dying. It is being maintained. Whether that constitutes a form of biological governance or something stranger still, the field has not quite agreed on what to call it. From the total colonization of an identity, we turn inland, to the meadows of temperate Europe, where a flatworm has engineered something arguably more audacious than any of these. It did not just take over one host, it built a trap that requires three. Dicrocolium dendriticum, the flatworm that designed its own three-host trap. Stand in a European meadow in early evening and watch the ants at the base of the grass. Most of them are doing what ants do, moving purposefully, carrying things, responding to colony signals. But some of them are climbing. Up the grass stems to the very tips and then stopping. Gripping the blade with their mandibles and freezing there, exposed against the fading sky until the temperature drops enough to release them. They do this every evening. They will do it again tomorrow. They are not doing it for themselves. There is a flatworm managing the schedule. Dicrocolium dendriticum, the lancet liver fluke, needs to reach the liver of a grazing mammal, a cow, a sheep, a deer, to complete its sexual reproduction. The path it has engineered to get there requires a land snail, a specific species of carpenter ant, and a behavior modification system so elaborate that when parasitologist Wendell Krull and his colleague Charles Mapes finally mapped the full life cycle in a 1952 paper in Cornell Veterinarian, it reportedly took the field some time to accept that a single organism had figured all of this out. The sequence runs as follows. Fluke eggs deposited in mammal feces are consumed by land snails. Inside the snail, the parasite develops into cercariae, larval forms, which the snail expels in sticky mucus packets called slime balls. Ants collect these packets, possibly mistaking them for food or moisture, and ingest them. Inside the ant, the cercariae migrate to the abdomen. All of them except one. One larva travels instead to the ant's subesophageal ganglion, the primary behavioral control node, and lodges there. It never goes to a mammal liver. It never reproduces. It sacrifices its entire reproductive future to become a biological steering mechanism for the 40 to 100 siblings traveling in the ant's abdomen, waiting for a grazing animal to swallow them. The brainworm, as it is informally known, induces the evening climbing behavior documented in field observations across Germany, Switzerland, and North America. Infected ants ascend grass tips at dusk and in the cool of early morning, gripping tight and remaining visible to grazing livestock. When midday heat arrives, the behavior ceases and the ants return to normal activity, protecting the parasites from lethal temperatures. It is a two-condition behavioral switch. Expose the host when the risk to the parasite is low, retract the host when the risk is high. Executed inside a creature the size of a pencil tip, using a brain the size of a grain of sand. Research by Maria Manga Gonzalez and colleagues documented the selective migration of the brainworm to the subesophageal ganglion specifically, not to adjacent neural tissue, not to any of the metabolically richer regions of the ant's body. The targeting is precise in a host species the parasite has never encountered during its own development inside a snail. And the temperature-dependent behavioral gate requires the parasite to sense ambient conditions and generate a conditional response, a logical operation in a flatworm larva, with no structure we recognize as a sensory organ adequate to the task. Mainstream parasitology explains multi-host life cycles as stepwise elaborations. An ancestral two-host fluke occasionally infected insects that mammals then ate. Strains producing mild behavioral changes in those insects spread more successfully. The brainworm sacrifice strategy emerged through kin selection, since the controlling larva shares genes with the siblings it enables. Over millions of generations, the specificity accrued. The temperature-dependent switch is explained as a byproduct of the parasite's own metabolic sensitivity to heat, a passive response that happens to produce the right behavior at the right time. But a passive metabolic response does not explain why the one brain worm navigates to the subesophageal ganglion rather than anywhere else in an ant it entered for the first time. It does not explain how the behavioral window was calibrated to the grazing schedules of ruminant mammals. And it does not fully dissolve the problem of kin selection sacrifice in an organism with no kin recognition system we have been able to identify. Dicrocolium raises a question that keeps surfacing across this entire list. When a system requires this many simultaneously correct features to function at all, what exactly does stepwise mean? From the European meadow and its evening grass tips, we plunge into water, where a parasite has taken the idea of behavioral override and stripped it to its most violent possible expression. Gordian worm, the suicide programmer. A cricket walks to the edge of a pond. Every instinct in its nervous system evolved over hundreds of millions of years to keep it alive on dry land, to keep it away from drowning, away from open water, away from the exposure and the cold, is telling it to stop. And the cricket walks in anyway. It goes under. Inside its body, coiled like a watch spring, taking up more space than the cricket's own organs, a worm 10 times the cricket's body length waits for the water to trigger its release. It will be out within 30 seconds. The cricket is not coming back up, and the worm has been doing this since before the first flower bloomed on Earth. The Nematomorpha, hairworms, or Gordian worms, named after the legendary knot, have a confirmed fossil record extending to Cretaceous amber specimens dated to approximately 100 million years ago, recovered from Myanmar deposits. Some phylogenetic analyses suggest the phylum's true origin may be deeper still, possibly Cambrian, though the amber record provides the most direct evidence we can point to confidently. What we can say with certainty is that the drowning compulsion they produce has been tested and refined across geological timescales against nervous systems that have themselves been evolving for just as long. And the compulsion still works. The mechanism was first characterized biochemically by a French team led by David Byron, published in Proceedings of the Royal Society B in 2005. The team used proteomics to identify which proteins Spinochordodes tellini, a hairworm infecting Mediterranean field crickets, was producing inside the cricket's nervous tissue. What they found was not a blunt chemical assault. The worm was synthesizing proteins that mimicked the cricket's own WNT signaling molecules with receptor-level specificity. WNT proteins are not obscure compounds. They are among the most conserved signaling molecules in the animal kingdom, present in organisms from flatworms to humans, governing the formation and function of neural circuits during development and beyond. The hairworm was not just drugging the cricket. It was rewriting the cricket's neural architecture using the cricket's own molecular language. A follow-up study in 2006 confirmed the same mechanism in a separate hairworm species infecting desert locusts. Creatures evolutionarily hardwired to avoid water. Same strategy, same molecular target, same outcome. The host walks into the water. The worm coils out through an opening in the exoskeleton within seconds of submersion, triggered by the contact with water acting as a final release signal. The timing is exact. The worm exits before the cricket stops moving. It then has hours, perhaps days, to find a mate in the water before its own reproductive window closes. The entire suicidal program is synchronized to the worm's reproductive maturity. It does not trigger early. It does not trigger late. It triggers once when it must. An adult Spinochordodes can reach 20 centimeters in length inside a cricket whose entire body spans 3 to 4 centimeters. It has consumed the cricket's fat reserves and most of its internal organs over weeks of development, leaving only the structures needed for locomotion, the legs, the exoskeleton, the minimal neural architecture required to receive commands. The cricket is, by the time it reaches the pond's edge, less a cricket than a remote-controlled chassis. Evolutionary biologists explain this as directional selection acting on a simple pressure. Hairworms that need water to reproduce. Any ancestral worm whose metabolic byproducts happen to push the host toward moisture transmitted more offspring. The WNT mimicry is explained as a possible product of horizontal gene transfer. The parasite may have incorporated host gene sequences over millions of generations of coevolution, not synthesizing the pathway from scratch, but repurposing stolen architecture. It is a more economical explanation than independent biosynthesis. It is also, in its own way, deeply strange. Because if the hairworm's behavioral control toolkit contains sequences borrowed from its host's own genome, then what the worm is using against the cricket is, in part, the cricket itself. The lock and the key made from the same material. The cricket is drowning in something it helped build. Some researchers studying nematomorph genetics have described this as one of the most vivid possible illustrations of how porous the boundary between parasite and host actually is. Not two separate organisms in conflict, but two genomes that have been exchanging material across deep time until it is no longer entirely clear where one ends and the other begins. From the violence of the drowning compulsion, we turn now to something more theatrical, a parasite that does not drive its host to destruction through brute neurochemical override. It does something far more elegant. It turns the host's most visible feature into a stage. Leucochloridium paradoxum, the parasite that turned eyes into puppets. Most parasites hide. They burrow into tissue, suppress immune responses, make themselves invisible to the host's defenses and to the predators that might notice something is wrong. Leucochloridium paradoxum does the opposite. It makes itself as visible as possible in the most conspicuous location available, and then it forces the host into the brightest, most exposed place it can find. Concealment is not the strategy. The strategy is spectacle. Leucochloridium is a flatworm that infects amber snails, small, moisture-loving creatures that live in the damp leaf litter and shade of European and North American woodlands. Amber snails avoid sunlight. Sunlight desiccates them, strips the moisture from their soft bodies, and exposes them to aerial predators. Their phototaxis, their instinctive movement away from light, is not a preference. It is a survival mechanism refined over millions of years. An infected amber snail moves toward the light. Not reluctantly, compulsively, but the behavioral inversion is only part of what makes Leucochloridium remarkable. Once inside the snail, the parasite develops into a sporocyst, a brood sac, and migrates selectively into the snail's eye stalks. Not into the mantle, not into the foot, not into any of the metabolically richer tissues it passes on the way. Into the eye stalks specifically. The eye stalks are transparent. They are thin-walled, and once the sporocyst is established inside them, it begins to pulse, contracting and expanding rhythmically, its banded coloration of green, yellow, and brown strobing through the translucent tissue. Research comparing the pulsation to the movement of caterpillars common to the same woodland habitat found the frequency and visual pattern to fall within the range of prey items that insectivorous birds actively target. The snail's eye stalk has been transformed into a lure. Bird swoops down, plucks off the pulsating eye stalk, swallows it, and with it, the sporocysts that will mature in the bird's intestine, producing eggs that are shed in feces, consumed by another snail, completing the cycle. A controlled observational study by Wesołowska and Wesołowski, published in the Journal of Zoology, documented that infected amber snails spent significantly more time in exposed sunlit areas than uninfected individuals, a behavioral reversal measured across enough specimens to be statistically robust rather than anecdotal. The light-seeking was not mild or occasional. It was consistent and directional, contrary to every behavioral tendency the snail would otherwise express. What makes the system particularly striking is that the snail survives the theft of its eye stalk. The amber snail regenerates the removed stalk over the course of several weeks, and the Leucochloridium infection remaining in the snail's body migrates into the new stalk and begins the pulsation cycle again. The snail can be harvested multiple times by multiple birds. It is less a dead end than a renewable resource. The mainstream explanation frames the light-seeking behavior as a consequence of the sporocyst's physical presence in the eye stalk pressing against the optic ganglion, disrupting phototactic processing, and effectively reversing the input-output relationship of the light-sensing circuit. The pulsation is the parasite's intrinsic metabolic rhythm, its own contractions made visible by the transparent tissue it occupies. The resemblance to a caterpillar is considered coincidental convergence, the kind of mimicry a moving object in a visually active predator's environment will naturally seem to produce, since birds are primed to investigate motion. But coincidental convergence does not explain why Leucochloridium migrates into the eye stalk specifically, a tissue with minimal nutritional value, rather than any other part of the snail's body. It does not explain why the pulsation rate across multiple geographically isolated populations on 2 continents falls consistently within the caterpillar mimicry range rather than at some random metabolic frequency. And it does not explain why the single most transparent, most exposed, most visible structure in the snail's body is also the one the parasite selects, in an animal the parasite cannot see and inside a system the parasite could not have surveyed before entering it. Some parasitologists describe Leucochloridium as a possible example of sensory exploitation, a parasite that has evolved to produce outputs that trigger pre-existing perceptual biases in predators it has never directly encountered. If so, it has calibrated that exploitation to a 2 mm window of anatomy and a specific frequency of movement inside a host it occupies blind. Whether that calibration is coincidental, converged, or something the field does not yet have a clean word for remains an open question. From the visible to the invisible now, from a parasite that makes itself spectacle to one that makes itself absence. One that is everywhere and nowhere, operating inside the most complex nervous system on the planet, and doing so in at least 1 in every 3 humans alive today. Toxoplasma gondii. The ancient parasite that may be inside your brain, right now. There is something statistically likely about the following statement. If you are watching this, there is roughly a 1 in 3 chance that what you are about to hear describes something currently happening inside your own skull. Not metaphorically, not as a thought experiment. A single-celled parasite, ancient enough to have been present when the first large mammals were still figuring out what the world looked like after the asteroid, may be living in cysts in your amygdala and prefrontal cortex right now. It formed those cysts quietly, without symptoms, possibly years ago. And the question that no researcher has yet closed is whether it has been doing nothing since then, or something we simply have not learned to measure correctly. Toxoplasma gondii was first isolated in 1908 from a North African rodent. Its name references that animal, Pterodactylus gundi. The parasite belongs to an ancient lineage estimated by molecular clock analyses to have diverged from its nearest relatives approximately 65 to 100 million years ago, coinciding broadly with the rise of the placental mammals it would come to inhabit. Its sexual reproduction occurs only in one place, the intestine of a cat. Every other warm-blooded vertebrate on Earth is an intermediate host, a vehicle the parasite needs to travel through to reach that intestine. To improve the probability of that journey completing, Toxoplasma does something that Joanne Webster at Oxford University first formally documented in the 1990s and published in landmark papers in 1994 and 2000. Infected rats lose their innate fear of cat odor, not fear in general. Their responses to other predators remain intact. Specifically, the olfactory fear response to felid scent is selectively suppressed. In some studies, infected rats appeared to develop a mild attraction to the scent. In Webster's controlled experiments, the behavioral difference between infected and uninfected rats was measurable, consistent, and reproducible. A parasite was editing a specific fear circuit in a vertebrate brain in the direction of making that vertebrate more likely to be eaten by the parasite's reproductive host. In 2011, Wendy Ingram at UCSF demonstrated that this effect persists even after the parasite is cleared from the body. The neural remodeling is permanent. The cysts restructure the circuitry and then leave the changes behind. An estimated 30 to 50% of the global human population tests seropositive for toxoplasma, according to surveys compiled by Pappas and colleagues in a 2009 bulletin of the World Health Organization. In some populations, parts of France, Brazil, rates reach 80%. These are chronic infections. The cysts in human brains form and remain largely without inflammation, largely without detectable immune response for decades. The brain does not clear them. It walls them off and leaves them there. Jaroslav Fleger at Charles University in Prague spent years testing whether seropositive humans show measurable behavioral differences from seronegative controls. His findings, published across multiple papers through the 2000s, identified elevated risk-taking behavior, marginally reduced reaction times, and elevated testosterone in seropositive men, alongside a different but also measurable personality profile in seropositive women. A 2002 Czech study found seropositive drivers approximately 2.5 times more likely to be involved in traffic accidents. Statistical associations between seropositivity and schizophrenia have been reported at roughly a 2-fold to 3-fold elevated risk across multiple independent datasets, though the causal pathway remains contested. The mainstream position is that human behavioral effects are incidental. Humans are evolutionary dead ends for toxoplasma, never eaten by cats, so any changes in us are spillover from a rodent targeting program operating in the wrong host. The cysts in human brains are doing nothing purposeful. We are just catching stray fire from a system that was never aimed at us. But toxoplasma encodes 2 functional tyrosine hydroxylase genes in its own genome, enzymes involved in dopamine synthesis that are active specifically inside brain cysts in mammalian hosts. Dopamine regulation in the prefrontal cortex has nothing to do with surviving in a cat intestine. It has everything to do with reward, risk, and behavioral salience in the host the parasite is currently occupying. A parasite that accidentally ended up in the human brain and is doing nothing purposeful, there does not typically pack dopamine synthesis enzymes in the luggage it brings along. The question some researchers find themselves unable to drop is not whether toxoplasma is changing human behavior. It is whether what we are currently able to measure is the entire effect, or only the parts we know to look for. The invisible now gives way to the architectural. A wasp in the forests of Central America has solved the problem of behavioral control not with chemistry, but with construction. It does not just steer, it builds a bodyguard. Glyptopantels, the wasp that turns a caterpillar into its own bodyguard. A caterpillar sits on a leaf covered in small white cocoons. The cocoons are the pupae of the wasps that just spent weeks eating it alive from the inside before cutting their way out through its skin. By any standard logic, the caterpillar should be dead or dying. Instead, it is thrashing. Every time something approaches the cocoons, a stink bug, a fly, a predatory insect, the caterpillar whips its body back and forth with enough force to send the attacker retreating. It is protecting the things that destroyed it, and inside its body, a few wasp larvae remain, still alive, never to emerge as adults. They are the engine keeping the bodyguard running. This is Glyptopantales, a parasitoid braconid wasp from the forests of Central America, first described behaviorally in a 2008 paper by Amir Grossman and colleagues at Wageningen University, published in PLOS One. The family Braconidae has a confirmed fossil record in Cretaceous amber dating to approximately 90 million years ago, placing the parasitoid lifestyle well within the age of dinosaurs. What Grossman's team documented in the Costa Rican forest was something the field had not previously quantified: the deliberate retention of larvae inside the host, after the majority had already emerged, for the sole purpose of generating and sustaining the caterpillar's defensive behavior. The life cycle moves in 2 distinct phases of manipulation. In the first phase, the female wasp injects dozens of eggs into a living caterpillar, typically a geometrid species such as Therantina leucocerae, using a hypodermic ovipositor in a strike so fast the caterpillar barely registers it. The larvae hatch and begin consuming the host's internal fat reserves over a period of approximately 2 weeks, carefully avoiding the organs needed to keep the caterpillar alive and mobile. When the larvae are ready to pupate, they chew through the caterpillar's skin simultaneously and form white cocoons on the leaf surface beside their host. The caterpillar is still alive. In the second phase, a subset of larvae, typically 2 to 4 individuals out of a cohort of 20 to 80, do not emerge. They remain inside the caterpillar's body, and the caterpillar, which had been moving and feeding normally throughout the first phase, now becomes almost entirely stationary, positioned directly adjacent to the cocoon cluster, and begins the violent defensive thrashing that Grossman's team documented in controlled predation trials. When the stink bug Podocyst nigrospinus was introduced in experimental conditions, caterpillars with internal larvae produced significantly more defensive head swinging than caterpillars from which the remaining larvae had been experimentally removed. The larvae are confirmed as the active behavioral driver. Without them, the bodyguard behavior ceases. The guardian larvae never complete their development. They remain as larvae until the adult wasps emerge from the cocoons and disperse, at which point the caterpillar dies. The guardians sacrifice their entire reproductive future for siblings they will never encounter as adults. Kin selection provides the theoretical framework. The guardians share genes with the pupating siblings, so behaviors that increase sibling survival increase the guardian's inclusive fitness even at the cost of direct reproduction. The logic is sound. What the logic requires, however, is that the remaining larvae inside the caterpillar can detect the approach of an external predator to external cocoons through the caterpillar's body wall in real time and translate that detection into behavioral commands the caterpillar's neuromuscular system then executes. The guardians are, in some functional sense, reading the caterpillar's sensory input and using the caterpillar's motor output as their own. The caterpillar has become a sensory relay station and a weapons platform simultaneously, operated remotely by organisms living inside it. Some entomologists studying Glyptopantales have described this as one of the clearest biological examples of an extended superorganism, a kin group that collectively occupies and operates a shared biological chassis. Others have noted that the system works too precisely to be a rough approximation. The defensive thrashing is not random agitation, it is targeted. It responds to approach. It stops when the threat retreats. We do not yet have a complete account of how the internal larvae detect external approach, what signal the caterpillar's nervous system is carrying that they intercept, or how the command for defensive thrashing is issued and received. The mechanism remains partially open. What is not open is the outcome. The bodyguard works, the cocoons survive at higher rates. And somewhere in the amber record, 90 million years back, ancestors of this wasp were already working out the early steps of a system that would eventually build itself a living fortress out of the body of the creature it consumed. From a bodyguard built from a living caterpillar, we turn now to the creature that haunted the most important biologist in history. The parasite that made Charles Darwin sit down and write a letter he could not stop writing, because what he was describing refused to fit inside a world made by a benevolent hand. Ichneumon wasp. The creature that made Darwin. Question God. In May of 1860, Charles Darwin wrote a letter to the American botanist Asa Gray that has never been fully answered. He had been thinking about the natural world, about design, about the argument that the complexity of life implied a creator who intended goodness. And he kept arriving at the same obstacle. He could not, he wrote, persuade himself that a God both benevolent and omnipotent would have designed a wasp with the express intention of feeding within the living bodies of caterpillars. The Ichneumon wasp was his example, not an edge case, not an obscure aberration. He was describing the most species-rich family of any organism on Earth. The Ichneumonidae contain an estimated 100,000 described species, with total diversity possibly reaching 300,000 or more, a number that exceeds the entire count of vertebrate species by a factor of roughly 20. Every single one of them is a parasitoid. They lay eggs in or on a living host. The larvae eat the host alive with a precision that preserves the vital organs until last, because fresh food keeps better than dead food. The host does not die until the larvae are ready for it to die. This is not incidental. It is the design of the thing. Fossil ichneumonids have been recovered from Cretaceous amber approximately 100 million years old, and phylogenetic analyses suggest the family's origin may reach back to the Jurassic over 145 million years ago. Darwin's letter references a general truth the fossil record confirms. This system has been operational across virtually every terrestrial ecosystem on Earth for longer than mammals have existed. The venom that makes it work is not a blunt instrument. Research into the neurotoxic chemistry of ichneummonid venoms has revealed highly specific cocktails targeting only the motor neurons responsible for host locomotion, immobilizing the host without inducing the physiological cascade of systemic death. The host is alive. It remains metabolically active, producing the fresh tissue the larvae require. It simply cannot move. Some hosts are maintained in this state for up to 4 weeks. The bone regrowth and cellular regeneration that would normally repair damage are still operating inside a body that cannot do anything about what is happening to it. Some ichneummonid species carry an additional tool that deserves its own moment of attention: polydenoviruses. These are DNA viruses fully integrated into the wasp's own chromosomes, not infections, not external agents, but heritable genetic material transmitted to all offspring, expressed specifically in the venom gland and injected with the egg into the host at the moment of oviposition. Their function is to suppress the host's immune system, preventing the egg from being encapsulated and destroyed by the host's own defenses. The virus was once a parasite of the wasp. Over the course of deep evolutionary time, some estimates place the integration event at roughly 100 million years ago. It became an organ, a tool, part of the wasp's own genetic identity. Among the wood-boring species, the ovipositor can drill through several centimeters of solid timber to reach a host larva hidden inside, guided by vibration sensing and chemical detection through the wood itself, locating a target smaller than a centimeter across with no visual access and no direct physical contact. The females of some species can assess host quality at the moment of oviposition and determine the sex of the egg they will deposit, selectively fertilizing or withholding fertilization to match the egg's sex to the resource available. Evolutionary biologists explain all of this through arms race dynamics. Every host that evolved resistance to blunt paralytic agents created selection pressure for more targeted venoms. Every egg that was encapsulated by an immune response created pressure for more effective immunosuppression. Over 100 million years of this ratchet, extraordinary specificity accumulated. The polydenovirus integration is rare but not unique. Examples of ancient viral sequences co-opted into host genome function appear across the tree of life. The ovipositor precision reflects refinement under selection in an organism whose entire reproductive fitness depends on hitting a hidden target. But the polydenovirus is not just a co-opted pathogen, it is a fully domesticated one, encoding proteins that are expressed in a specific gland, injected at a specific moment, and perform a specific function that the wasp could not perform on its own. The parasite became an organ, the alien became self, and that transition, from enemy to tool, from infection to inheritance, raises a question that the ichneumon wasp leaves open long after Darwin's letter closes. How much of what any organism calls itself was, at some earlier moment, something that arrived from outside? From the wasp and its ancient domesticated virus, we go back, much further back than any of the organisms we have visited, to a parasite so old that it was already refining its behavioral toolkit before fish existed, before land animals existed, before anything with a spine had taken its first breath. To the organism that may have written the first manual for stealing a nervous system 500 million years ago. Acanthocephala, the 500 million-year-old puppeteer. Before fish, before insects, before the first creature left the water and felt air on its body for the first time, before almost everything we would recognize as complex animal life had yet appeared. Something was already learning how to hijack a nervous system. The spiny-headed worms, the phylum Acanthocephala, have molecular clock estimates placing their divergence from related lineages at approximately 500 to 600 million years ago, in the Cambrian or possibly the Ediacaran period. They have been doing one thing across all of that time: getting inside an animal, taking hold of its behavior, steering it toward death. They were here before the nervous systems they exploit had fully finished evolving. They may have been shaping those nervous systems from the beginning, exerting selection pressure on the very architecture they would later learn to rewire. The mechanism was first quantified rigorously by Robert Paulin at the University of Otago, one of the most influential parasitology researchers of the modern era, in foundational papers published in Behavioral Ecology in 1994 and 1995. Paulin was studying Gammarus pulex, small freshwater crustaceans called amphipods infected with Pomphorhynchus laevis, an acanthocephalan. What he measured was not a single behavioral change. It was the simultaneous reversal of multiple independent behavioral systems, all in the same direction, all increasing the probability that the amphipod would be eaten by a predatory fish, which is the parasite's definitive host, the one place it can complete its sexual reproduction. Uninfected Gammarus avoid light, infected Gammarus seek it, spending nearly 4 times as much time in illuminated areas in controlled conditions. Uninfected Gammarus cling to surfaces and prefer the deeper protected regions of the water column. Infected Gammarus swim exposed near the surface. Uninfected Gammarus respond to predator-simulating vibrations by retreating. Infected Gammarus approach the source. These are not related behaviors. They are governed by distinct neural circuits mediated by different signaling pathways, each representing a separate evolved response to a separate category of predation risk. The Acanthocephala has reversed all of them simultaneously in a coordinated direction. The amphipod has been turned into something that moves through the water as if it is trying to be eaten. Research by Tayin and colleagues, published in 2006, found significantly elevated serotonin levels in the central nervous systems of infected amphipods compared to uninfected controls. Serotonin modulates locomotor behavior in crustaceans as it does in many animals, and the hypothesis that the acanthocephalan is producing a broad serotonergic elevation which then cascades through multiple behavioral circuits simultaneously is the mainstream mechanistic explanation. It is more parsimonious than imagining the parasite has separately targeted each behavioral system in sequence. One lever, many downstream effects. But serotonin elevation in a crustacean nervous system does not uniformly reverse all behaviors in the direction of predation. Different circuits respond to serotonin differently depending on receptor subtype, anatomical location, and behavioral context. The fact that the reversal is coordinated, that light-seeking, surface swimming, and vibration approach all shift together, all toward exposure, all toward the fish, implies either that there is a single master circuit governing all predation risk behavior in these animals that no one has yet identified, or that the acanthocephalan's intervention is more targeted than a simple serotonin flood. Both possibilities are remarkable in their own ways. The acanthocephalan has no digestive system of its own. It absorbs nutrients directly through its body wall from the host's intestinal contents. It is completely dependent on the host continuing to feed and digest. It maintains this dependence for what can be the majority of the host's remaining lifespan. and it accomplishes its behavioral override with a body plan so stable that it has not fundamentally changed since the Cambrian, a worm with a spiny proboscis, no brain worth measuring, and 500 million years of practice at the one thing it needs to do. We have built neuroscience, we have mapped the human connectome, developed psychopharmacology, engineered molecules that target specific receptor subtypes in the human brain with extraordinary precision. We developed all of that in the last century or two. The acanthocephala had a working version 500 million years before we started. 10 parasites, 10 different strategies, 10 different geological timescales, and one pattern running through all of them that is impossible to unsee once you have looked at the full list. Each of these organisms navigates a nervous system it has never seen, produces behavioral outcomes in a host that is not itself, and does so with a specificity that scales in many cases beyond what we would expect from a rough evolutionary approximation. The fungus calibrates to a 2-centimeter humidity band. The flatworm finds the subesophageal ganglion in an ant it entered through a snail. The hairworm synthesizes the WNT pathway proteins that speak to the exact receptor architecture of an insect brain. The barnacle finds the lingual artery in a fish and severs it before taking the tongue's place. These are not accidents that accumulated into functionality. They are solutions, precise ones. The mainstream scientific framework for all of it is evolution by natural selection, and that framework is not wrong. It is the best explanation we have, and it accounts for the directionality and the specificity of what we see in a way no alternative currently matches. What selection can do, given enough time and enough generations and enough pressure, is genuinely extraordinary. The 10 cases in this video are evidence of that, but they are also evidence that extraordinary may be underselling it. Some researchers studying parasitic manipulation have begun asking whether the conserved molecular architecture of animal nervous systems, the serotonin pathways, the WNT signals, the dopaminergic circuits, the EC-dysone cascades, is so deeply stable across hundreds of millions of years of host evolution that independent parasitic lineages keep finding the same entry points over and over, not because they copied each other, but because there are only so many keys, and the locks have not changed. If true, that would mean the biochemical lever points in animal brains are predictable enough to be repeatedly rediscovered by separate evolutionary lineages operating across geological timescales. That is a remarkable statement about the structure of neural architecture. It may be the most unsettling thing in this entire list. Most people look at a cricket drowning itself, at a crab tending someone else's eggs, at an ant biting a leaf at exactly the right height, and they see weird nature. They accept the textbook explanation and move on. But you made it here. You noticed that the hairworm carries the host's own molecular vocabulary. You noticed that toxoplasma packed dopamine synthesis enzymes for a trip through a human brain. You noticed that the acanthocephala was already doing this before vertebrates existed. That is not a passive consumption of information. That is analysis. And the question you are now sitting with, whether precision this deep across timescales this long in organisms with no brains to plan with, implies something about the nature of biological information that our current frameworks have not fully described. That question does not have a closed answer yet, which means it is still yours to work on. This channel is for the people who cannot leave that kind of question on the table. Hit subscribe and make it official. Then go to the comments and tell me which of these 10 cases feels least like a simple accident and most like something we do not yet have a name for. Make your case. The floor is yours. They were here before the fish, before the forests, Before the first nervous system had finished wiring itself together, something was already studying the blueprint. We look at these parasites and reach for words like primitive, like simple, like ancient, as if age were the same as limitation. But 500 million years of practice at one task looks nothing like primitive. It looks like the zombie ant biting at exactly the right height in the right forest at the right time of day. It looks like the crab that forgot it was a crab, tending eggs that are not its own with the full devotion of a parent. Whatever these organisms know, and whether knowing is even the right word for what they do, they knew the shape of a nervous system long before we had the language to describe one. Subscribe, because this is only the beginning.